Thank you for that warm introduction. So, yes, there are a lot of results, and so there's a lot of material to get through, so let's roll right into it. Because there's so much, I'd like to provide you an outline. As many presentations do, I'll start with an introduction, move into the methods, start with my first study, the methods comparison results. We'll discuss the methods comparison. We'll pursue the results for the habitat transition study, discuss those results, and then wrap it up with a final takeaway of both of these studies. So, why should we or why do we care about kelp forests? Well, they're beautiful, and I get to spend almost every day in them, which is very fortunate for. They support one of the most diverse ecosystems in the world and are financially very productive. Kelp forests globally support a $500 billion industry through fisheries production and nitrogen removal. And within the California current, which is where we are residing right now, these kelp forests generate about $50 billion a year. Giant kelp alone generates about $35 billion, which is the most common canopy farming kelp that we have here in Monterey. And in order to protect these resources within and around kelp forests, California, has established a massive MPA network. This network was revamped and expanded in 2012 and now encompasses over 800 square miles of protected waters that cover many different habitats, including kelp forests. These MPAs require a vast amount of monitoring so that we can determine the influence and efficacy of the different types of closures, the different effects that these closures have on these habitats. Unfortunately, California and other regions around the world have begun experiencing widespread kelp forest declines. Along North and Central California, we've seen declines in canopy forming kelp of up to 90%. And this figure from Smith et al. Really wonderfully demonstrates the resulting effects, namely in urchin behavior of this kelp forest fallout that started in 2014. As algae stocks decline, urchins emerge from their crevices, changing from passively foraging on drift algae to actively grazing on standing kelp. This destroys the drift algae production and creates a positive feedback loop where these urchins are consistently and persistently actively grazing down drift algae or standing algae. A significant part of the urchin-kelp relationship is the simple fact that they share an association for hard and consolidated substrates. Urchins must be able to reach the standing kelp in order to graze it down, and to do this they form destructive fronts. However, we often see kelp beds and urchin barrens meeting and these boundaries persisting for considerable amounts of time. So why are urchins not advancing through these boundaries at these kelp bed edges? One answer could be that certain substrates, such as sand, act as barriers to urchin movement. Lohr et al., way back in 1986, examined the hesitancy and reluctancy of purple urchins specifically to navigate across sand channels in search of food. Comparatively, red urchins didn't demonstrate such a strong hesitancy. Another contributing factor to urchin barren and kelp forest boundaries may be algae abrasion, so the physical act of the algae moving against the urchins and deterring their movement into the kelp bed. So now that we've established a bit of a background on kelp forests and how urchins structure them through grazing, I want to discuss how we monitor these environments. So scuba diving as a subtidal monitoring tool has only really been around since the 1940s, and it was largely pioneered by these two gentlemen, Conrad Limbaugh and Wheeler North, hailing out of Scripps down in San Diego. And since their time, just less than 100 years ago, there are hundreds, thousands more likely, of subtidal surveying programs and protocols all over the world. One of the most common protocols is the Underwater Visual Census Survey. It's a relatively straightforward and simple approach that has the diver count their target organisms and measure objects or species along or within a known area, typically along a transect line, like you see here. But not all areas of interest are surveyable by scuba divers. Deep sea ROVs are especially useful since we can access depths far beyond what scuba divers can reasonably spend time at. They can also be outfitted with various forms of instrumentation, like the green lasers that you see in this image. These lasers are paired and set a known distance apart. I believe these ones are 10 centimeters apart. And because we know that distance, we can measure things in this field of view so long as they are on the same plane. When things start to be angled relative to the camera, these measurements can get a little complicated. So this brings me to my tool of choice, diver-operated stereo video. So stereo video allows us to do many of the same things that diver UVC protocols do, but with the added measurement precision, while retaining some of the benefits of having a diver actively in the water. These two rigs are the rigs that I used throughout my thesis. They both have two cameras in waterproof housings that are angled slightly in towards one another to create an overlapping field of view. And if anybody's curious on these size differences, the top rig weighed about 35 pounds and that bottom rig weighed about 5 pounds. And the transition to that smaller one was really, really welcome, considering we do primarily shore diving here. I only did shore diving for this. So this is an example of the left and right perspectives. And by identifying the same points and same areas in the two perspectives, we establish this shared overlapping field of view in which we can derive 3D measurements from two-dimensional images. And this can be made of pretty much any object that's in this field of view. However, perspective is really important when we're using stereo video. This can lead to different measurements depending on the target and the perspective that you adopt. So Bennett et al. Compared forward and downward facing perspectives in a coral reef environment and found that percent cover measurements using forward facing perspectives produce greater estimates of vertical growing organisms like those branching corals up on the top, those top three. Similarly, horizontal growing organisms and various forms of benthic habitat, percent cover measurements were greater using downward facing perspectives relative to forward facing perspectives. So angle of approach matters depending on your target, whether it's habitat or species. So we covered a few of the traits of stereo DOV and diver visual surveys, but before we continue and get into my research questions, I want to cover their trade-offs more specifically. Imagery in general facilitates rapid data collection. We're not having to note things down by hand as often. This can allow surveyors to cover much larger areas. As I mentioned before, stereo DOV gives us the ability to precisely measure things. The data are reviewable, which is really handy if you need to audit the data or use it as a training material. But the imagery often comes at the cost of reduced taxonomic resolution. For these diver UVC surveys, we benefit from their generally simple technology. In that photo you saw a slate, a pencil, underwater piece of paper, and a transect tape. Not a whole lot of things to go wrong down there compared to bringing expensive electronics. Because of the simplicity and the fact that the diver is responsible for so much of the data collection down there, this approach is really flexible to a number of different applications and environments, but it often does require the surveyor to not only be a competent and capable diver, but they also have to be an expert of the system that they're studying, or at least the protocols that they're employing. And then, of course, as with any scuba diving project, especially those that aren't venturing into the technical side of things, there are pretty restrictive depth and time limitations. However, despite all this that we know, there are still questions that remain for specific applications of stereo video. So, understanding that, my first research interest that I'll be discussing is how diver-operated stereo video and diver underwater visual census surveys compare when performing benthic surveys in kelp forests. To do this, my plan was to use two different long-term subtidal surveying protocols. The first was the Partnership for Interdisciplinary Studies of Coastal Oceans, otherwise known as PISCO, and their protocol for Uniform Point Contact, which is a habitat classification protocol. And Reef Check California's swath, invertebrate, and algae protocols for swath and for algae and inverts. PISCO is comprised of largely academic institutions and people with a fair amount of taxonomic familiarity with the system. And so they have a much finer demand for taxonomic resolution than Reef Check California, which leverages citizen science to scale up their monitoring approach. So anticipating the reduced taxonomic resolution of the imagery that I was exploring, I opted to use Reef Check's relatively simpler list. This is a list of the common species, common target species that I was interested in. The list that I was using was much larger, but these are generally the species that we reliably find in the Monterey Bay. And some of these are particularly important for kelp forest regimes, and this will be more important for the habitat transition component, but these are the species that I gave a little bit more attention to. So, moving on to the research questions and my specific hypotheses. First, concerning diver-operated stereo video compared to diver UVC, I hypothesized that community metrics such as diversity, richness, and density made from UVC surveys would be greater than those produced using stereo DOV. That diver UPC and stereo DOV habitat surveys would produce similar habitat characteristics of relief, substrate, and cover, and I'll describe those in more detail later. And then I was also interested in exploring the relationship between a 45-degree angle of approach and the 90-degree downward-facing angle of approach. And I hypothesized that the downward-facing approach would produce similar benthic macroinvertebrate estimates to UVC swath surveys. And the last hypothesis for the methods comparison was exploring the effect of algae density on stereo DOV performance. And I hypothesized that stereo DOV performance would be degraded relative to UVC estimates as algae density increased. So the second component of my thesis was concerned with habitat transitions, and this will get more attention in a little bit. But my objective for this one was to use stereo video to evaluate the influence of habitat transitions on urchin and kelp populations using the same PISCO and Reef Check protocols that I explored in the methods comparison. My hypotheses for these were far fewer, the first of which being that purple urchin densities would be negatively associated with substrate and relief transitions. Algae densities would be positively associated with substrate and relief transitions, and that red urchin densities would be negatively associated with relief transitions, calling back to that Laura et al. Study that found that red urchins were more readily able to cross sand. Okay, on to the methods. And this is the view that those urchins got for 3 years. So all of my survey sites were located in Monterey and Carmel Bays between depths of 3 to 15 meters. We surveyed San Carlos Beach, which many of you know as the breakwater. We surveyed the Wall and Middle Reef. We surveyed Maccabee Beach, Lover's Point, Monastery North, and Monastery South. And these sites were selected because most of them, they're all rocky reefs. Most of them are a kelp forest or mixed kelp forest urchin barren state, with the exception of Lover's Point, which is strictly an urchin barren. South Monastery has wide swaths of urchin barrens that are targetable, but it still has some kelp. So every single survey that I performed for both of my studies started more or less the same. One diver would reel out the transect tape, and the stereo video diver would follow, recording the benthos using whichever 45 or 90 degree protocol was called for at that time. And this process could take between 2 to 5 minutes. Again, I used 2 different surveying protocols, macroinvertebrate and macroalgae swath, which is outlined, the survey area is outlined in the blue there. And we made 2 passes, one at 45 degrees and one at 90 degrees. The other protocol was our habitat characterization protocol, UPC. This used a narrower survey area and used just 1 pass at a variable 45 to 90 degree angle. So it wasn't part of the angle comparison component. The swath surveys were conducted over a 10-meter transect, 1 meter to either side. And this 10-meter transect would take between 5 and 10 minutes to complete just for the swath surveyor, so just for the diver that was down there, not the stereo video flyer. The swath surveyor would count all macroinvertebrates within the survey area greater than 2.5 centimeters. They would count all stipitate algae, which from now and moving forward is going to refer to all macroalgae that I'm concerned with the exception of giant kelp, greater than 30 centimeters. And then giant kelp, Macrocystis pyrifera, greater than 1 meter, taller than 1 meter. For the UPC protocol, again, it was a smaller survey area. These surveys were done on a 30-meter transect because we could do them quicker. Time was a big consideration for these surveys. And these usually took between 12 and 15 minutes. Unlike swath, which is 1 meter to either side of the entire transect, UPC surveyed a 1/4 meter in front of and behind each meter point and 1/2 meter to either side, producing 30 half-meter squared quadrats. Relief was the first component of habitat that was measured. Relief is the measurement from the highest to the lowest point within the survey area. And this was binned into 4 categories ranging from less than 10 centimeters to greater than 2 meters. Substrate category was classified as the size of the habitat directly below each meter point, again binned into one of 4 groups. And the last was benthic cover, which is slightly different than the rest. It is a semi-random categorization of whatever is growing or occupying the substrate. So the diver would close their eyes or look away, and after making sure the point was safe, throw their finger down and record what their finger touched. This could be various forms of algae, different sessile invertebrates, or one of our few inanimate categories like bare rock or bare sand. For the transition study, we again rolled out that transect tape. This one was 100 meters since there was no swath or habitat characterization happening underwater. And only one angle was used, for a variable flight angle similar to UPC, 45 to 90 degrees, depending on the features of the benthos that we were encountering. And for analysis, we extracted 3 random 10-meter segments from the 100-meter transect. So this is Event Measure. It's made by an Australian company, CGIS. And this is where I could extract all of my data. This is where I made my measurements and conducted my point counts of all the species. And I really like this frame because it highlights one of the less discussed benefits of using stereo video. There is a bat star that you can see in the left perspective that is obscured from the right perspective. And so if we were using a single perspective lens and the right perspective was all we had, we would not be able to see that species, organism. So for the swath surveys, data extraction. So this is now we're out of the water, behind the computer, doing these surveys all over again, but using stereo video. I would measure 1 meter to either side of the transect tape to establish my surveying area for that frame, counting every single organism within that frame. So this was at South Monastery inside the Urchin Barrens. So there are a ton of purple urchins. I did no subsampling. The only measurements that I made of these organisms was to confirm that they were large enough to meet my threshold for counting. And this process, again, on a 10-meter transect could take anywhere between 15 minutes to 60 minutes. For the habitat UPC stereo video data extraction, I would similarly measure out my survey area, and then I would visually identify the greatest relief feature and do my best to measure it. And we can talk more about the nuances of that if you're interested. And then I would measure the substrate directly below the meter point, as well as semi-randomly select and identify the cover category that was beneath that meter point. And this process could take between 20 and 60 minutes as well. Okay, so on to the results for the methods comparison. So for my analyses, I used a repeated measure linear mixed effects model, testing for interactions between survey type and reef type. And for the UPC comparison, I did not identify between kelp forest and urchin barren, so there's no test for interactions, and just testing for differences between the two survey types. So this is an NMDS plot, and really all you need to know is that this is a really good way to visualize the relationship between the community structure found between these three survey methods. So this considers the number of unique species and how many of them there were, and plots them in this two-dimensional plane. And what we can see within urchin barrens is that there is a lot of overlap between all of these dots in these color-coded ellipses. So we see almost identical overlap between 45 and 90-degree stereo video, and a considerable overlap with the swath invertebrate methods. And what this tells us, and what the permutational ANOVA confirmed, is that there were no significant differences in the community structure measurements made by either of our surveying approaches. There were, however, missing species from these stereo video approaches: rock scallops, ochre sea stars, rock crabs, and giant keyhole limpets. When we do the same analysis and visualization for the kelp forest community, we see, again, a lot of overlap between the 45 and 90-degree approaches, but swath has now kind of detached itself from this clustering, identifying itself as measuring a different, significantly different community structure. And this is more or less corroborated by the missing species list in kelp forests by stereo video, where we have a considerable number more. We added California sea cucumbers to that, 2 species of abalone, and another species of crab and red urchins. So continuing the theme of exploring community structure, this is a box plot where we have on our y-axis our measurements of species richness on the top there, which is the number of unique species found in the system. And then on the bottom y-axis, Shannon diversity, which is, again, a way to characterize community composition. How many unique species are there, and how evenly distributed are the abundances of these species across those unique species? So starting with urchin barrens on the left-hand side, similar to our NMDS plot analysis, I found no significant differences in species richness or Shannon-Weiner diversity by either of the 3 surveying methods. In kelp forests, this trend continues. I found 200% greater species richness using swath compared to 45-degree video, and 150% greater than 90-degree video. We also see significant differences between 90-degree video, which is that benthic downward-facing perspective, compared to the 45-degree video. And the story continues for diversity, finding very similar relationships where swath estimated the greatest diversity, followed by 90 degrees, and then lastly, 45 degrees. And so we're seeing a recurring theme of similar performance in urchin barrens and differing performance in kelp forests. So to explore this relationship further, I plotted swath-derived algae density on the x-axis there, and our various response variables on the y-axis to see how these metrics of interest change as algae density increases. On the top, we have species richness, and again, we already know that these two differ in urchin barrens, but we can see that as algae increases, these two slopes start to separate from one another. So you can see as algae density increases by 1/10 of a meter squared, richness reduces for our 2 video methods by 15%, and swath increased by 2%. So we're having a pretty distinct separation here. The story, again, continues for diversity, where we see reduction of about 1% in our video methods, but an increase in 2.5% for our swath methods. Now, this one, this is no urchin densities. So I removed urchin densities from this analysis because there was an overwhelming number of them present within the community. And so when we remove them from the analysis, we see, again, the story continuing, reduction in relative performance of stereo video compared to our diver swath estimates. However, when we only look at urchins, we don't see a difference in this relationship, where all 3 of them decreased by a relatively similar amount, around 70 to 80%. So remembering how important urchins are to this kelp forest story, I wanted to explore specifically how urchin densities changed in urchin barrens and kelp forests, how our estimates of them changed. With our stereo video methods, we see the greatest estimates being measured by swath invertebrate and 90-degree stereo video. So the differing letters indicate differences significant differences. But if they share any of the same letters, it means that there was no significant difference. So while swath was not different from 90, it did produce greater estimates compared to 45 degrees, but 90 degrees did not produce different estimates compared to 45 degrees. In the kelp forest, we see there's some dramatic numbers here. Swath estimates produced 1,700 times more urchins than 45 degrees, and 170 times more urchins than 45 degrees, or than 90 degrees. And these numbers are wild because, I think I found maybe like 2 or 3 urchins during these surveys. The last part of the methods comparison was examining habitat characterization. So this plot here, we have cover, substrate, and relief, and on the x-axis is the percent agreement, paired percent agreement between the methods. So we see that cover had about a 50% paired point agreement, meaning that the point that I identified through stereo video and the point that I identified underwater using the diver UPC protocols agreed about 50% of the time. For substrate, this agreement was slightly greater at 60%. And for relief, it had the greatest agreement at 75%. And without going too far into what the test statistics mean, it's a way to assess how strongly these two methods agree, taking into account the chance that it could randomly happen, right? So relief has a lower kappa test despite having a higher percent agreement because of the overwhelming presence of one of the relief categories. And if you want to explore this with me more, we can, but I'm not going to do that here. So we're going to move right on. All right, so let's discuss those results. If I could sum up these results in just one sentence, it would simply be that stereo DOV performs more similar to diver UVC in urchin barrens, and that algae is a significant predictor of stereo DOV performance. Missing species drove a lot of the differences that I saw between 45 and 90-degree stereo DOV surveys with respect to diver surveys, especially in kelp forests. So we identified in urchin barrens 11 unique species, 4 of which were missing from the stereo video surveys, and in kelp forests identified 15 unique species, 7 of which were missing. And what most of these species have in common is that they are cryptic, whether in behavior or the camouflage or their morphological structure. Other studies that have examined the difference in species richness between diver-operated video and diver visual surveys routinely find a reduction in taxonomic resolution that contributes to reduced species richness and Shannon diversity when using imagery techniques. And I found consensus for these previous findings in kelp forests, but not in urchin barrens. And this is likely due to the simple fact that algae just gets in the way. With respect to the relationship that we saw for urchins, they conveniently adopt an active grazing behavior in urchin barrens, making them easy to identify and resolve through video. But in kelp forests, if they are receiving an ample supply of drift algae, they'll adopt their crevice-seeking behavior, making them difficult to resolve through stereo video, but also more difficult to resolve for the divers. Regarding the performance as algae increases, a study conducted by Ling et al. Using autonomous underwater vehicles equipped with stereo video found a 40% reduction in urchin density estimates relative to what diver UVC measurements were as algae cover increased. So we did not see such a relationship. Remember, we found no significant difference in the slope performance of these methods. And I believe that one reason for the difference may be that the approach using a diver allowed for me to move algae out of the way, move around it if necessary. Remember, giant kelp extends all the way throughout the water column, so sometimes it was necessary to physically move this out of the way. And so by removing the offending algae, we could clear up our view of the benthos. This was not available to Ling's study using an autonomous vehicle. And for the habitat comparisons, our findings resembled those found by others, where diver estimated relief measurements were similar to those using stereo techniques or even just visual estimates through video. While I didn't explore our paired comparisons much in this presentation, I do want to recognize that I attribute a lot of the agreement, the percent agreement that we see there, to the large classification bins that UPC protocols use, which granted a significant buffer to any of the errors that may have been introduced. So I mentioned throughout the methods portion how long each aspect of this process took. And although this was not an explicit part of my thesis, the information I think is really valuable and important to consider when assessing the usefulness of either of these techniques. So considering how long it took to perform the underwater portion and the data processing portion, I found that diver swath surveys took, and this is all extrapolated out for a 30-meter transect, took about 20 to 35 minutes, while stereo swath surveys took about 50 to 181 minutes. Diver UPC took 17 to about 20 minutes, and stereo UPC took about 25 to 65 minutes. So consistently across the board, we are putting more time potentially into these imagery techniques than we would be spending underwater. So putting all of these comparison results together to establish a recommendation regime is very complex and very much based on many different factors, but this is my attempt based on a review of the literature and the results that I presented to you all here. Essentially, it comes down to the obstructions present in your habitat, the cryptic nature of your target organisms, for example, abalone, and how important species-level classification is for you, so your taxonomic resolution goal, how many people you have access to. Imagery oftentimes can be conducted with fewer people in the water. How much time you have, how important it is, how large is your data collection window. These all will go into informing which of these approaches may be most appropriate. Okay, so we move on to the habitat transition results. Take a sip of water here. So just a little briefing on this before we get into the results. I set out to measure complexity as changes in habitat structure. And so this can adopt one of four styles. We can have changes in substrate, where you see as we move along the transect, the benthos is alternating between sand and boulder or consolidated substrate. We can have changes in relief, where on the right-hand side, there is a massive, very tall wall compared to the relatively low relief portion of the boulders underneath it. There can be changes in relief and substrate, where flat, sandy habitats transition into taller, consolidated substrates. Or we can have relatively consistent habitats. We don't see changes in relief or structure like this cobble habitat. To calculate transition frequency, which I ultimately ended up using for the analysis, and to better capture the influence of these changes, I simplified substrate into a binary classification, optimal and suboptimal. Optimal are substrates that are more commonly associated with urchins and kelp, being boulder and bedrock, and suboptimal are those that are not commonly associated with urchins and kelp, sand and cobble. I did the same thing for my relief classifications, looking at flat relief, which is 0 to 10 centimeters, and not flat relief, which was greater than 10 centimeters. And so this yellow line here is a mock transect to give you an idea of how I calculated these habitat transitions in case it's not clear. Anytime a substrate category, for example, oscillated between optimal or suboptimal, the transition tick went up. And so in this 10-meter example, we have a total of 4 substrate transitions over the course of 10 meters. And for my analysis, I further simplified these transitions into 0 or 1 or more transitions for substrate and for relief, and I bifurcated depth into shallower than 12 meters or greater than 12 meters. And these binary categorizations were based on the ecological implications that these substrate and relief categories are expected to have on urchins and kelp. They also strengthened my model performance and improved fit, and only relief or substrate transitions were used in a final model. They never co-occurred in any of my models. So starting with our favorite benthic grazer, the purple urchin. On the y-axis, we have densities. On the x-axis, we have our shallow and deep categories. No transitions are in gold, and 1 or more transitions are in light green. And we can see that, that habitats with no relief transitions at depths shallower than 12 meters were associated with over 200% more urchins than those that had 1 or more transitions. And while not significant, this relationship was not seen at depths greater than 12 meters. In fact, we see the opposite. For red urchins, relief was also a part of their best fit model, but it did not produce any significant results. We did see an effect of depth with greater red urchins at depths greater than 12 meters. And for giant kelp adults, which is the final target species in the relief transition model, I found that at shallow depths, habitats with 1 or more transitions supported 100% more giant kelp adults compared to habitats with no relief transitions. However, these were only marginally significant, right? These alpha value of 0.05. We found it at 0.06 here. So moving on to the organisms that were associated with substrate transitions, giant kelp stipe densities did not yield any significant results with transitions or depth, and we don't see any strong relationships there. For stipitate algae, which again is all of our macroalgae other than giant kelp, we found that habitats with 1 or more substrate transitions supported algae densities up to 150% greater. And while the model suggested that depth did not interact significantly with substrate transitions, we do see a similar trend like we did with the urchin model, where the effect of transitions are not seen at deeper depths. Okay, so now let's go over those results. So again, if I could sum this up in one succinct sentence for you, it would be that simply habitat transitions generally, relief or substrate, were associated with fewer urchins and more algae. So what's the usefulness of this information? How do we use this? As I mentioned in the beginning, California and globally kelp forests are in decline. And lots of conversations are had around the world about what to do. And some of the most active discussions involve intervention and restoration. This wonderful paper from Geraldo Ospina from just January of this year looked at kelp canopy cover stability before the 2014 heat wave, which again contributed to mass kelp forest loss, during the heat wave and after the heat wave. And they used these relative abundances to classify these kelp forests based on their likelihood to accept restoration efforts. So we can see here in Monterey, we have a lot of high priority sites and a few very low sites. The high priority sites were identified as having a stable, healthy kelp forest before the event and a strong response after the event to recovery. So these authors did a wonderful job classifying all of these habitats, but they did not answer what makes them stable. What are the underlying benthic features or structures that contribute to kelp forest stability? And they recommended that people looking to use this resource study these sites to understand the mechanisms of resilience to the marine heat wave. And so that begs the question of what contributes to resilience. So complexity gets thrown into this part of the discussion more and more often. And this is one of my favorite studies that came out during my thesis from Randall et al. Back in 2022 from the Channel Islands. And what Randall and colleagues found was that over time, kelp forest stability, which on the, if we look at plot A, we have algae, mixed, and barren. Over time, kelp forest stability, which we can track as the likelihood that these points bounce between the algae and barren state, depended and was heavily influenced by substrate complexity. So as we increase substrate complexity, these sites, which are more complex on the right-hand side, look at West Dutch, oscillate far less often between algae and barren states, instead staying at a sustainable and stable mixed rocky reef state. Oops. So they measured complexity as rugosity. I measured it as habitat transitions, two different kinds, relief and substrate. But I must acknowledge that my two predictors did correlate quite a bit, bringing into question if there is a difference in the influence of substrate and relief transitions. And so this contingency table kind of shows you where these binary classifications fell and where they were associated. And one of the ones that I would like to point out is on the bottom one, we have substrate transitions, none, so a uniform substrate habitat, rarely ever correlated with relief transitions, meaning that if we saw substrate transitions, we probably also saw relief transitions. So, nevertheless, I believe that I have found evidence to suggest that habitat transitions using either relief or substrate may confer similar refugia to algae among urchins and other forms, like other forms of complexity have in previous studies. And based on my time studying these habitats, I believe that most of the resilient refugia that we've identified may exist more frequently on isolated reef patches among sand. So, the final takeaway bringing in these two projects together, when comparing stereo DOV and UVC, it's apparent that the demands of the study or project will largely determine stereo DOV's feasibility. Urchin barrens are likely an ideal habitat to apply benthic stereo video methods, especially if you're interested in broad-scale community changes. Habitat classification is possible and potentially comparable to diver-derived classification with considerations for the taxonomic desires of substrate cover. One thing I didn't have time to talk about was the simplification of these cover categories for the habitat classification. We can talk about that later if you want. When considering the influence of habitat transitions, I found that they were associated with more kelp and fewer urchins at depths shallower than 12 meters, and that substrate and relief may capture similar effects, at least in the way that I used them. So, as for next steps, what to do with this information, I would like to see fine-scale taxonomic assessments of stereo DOV surveys in urchin barrens. I would also like to see manipulative studies of urchins and kelp on different substrate and relief mosaics, and further study of the benthic contributors to kelp forest resilience. I think we're getting really close to figuring out what benthic features contribute to kelp forest stability, and those that will receive restoration and intervention initiatives well, but I don't think we quite have it figured out. Okay, so with that, I want to thank a number of people in my life who have contributed to this thesis, starting with the various departments, agencies, entities that have funded me, NOAA, EPP, MSI, CCME, and the GFP. These programs were instrumental in getting my methods comparison work completed. Without the support from these projects, I probably wouldn't have had the time to dedicate to it, and I might have finished a year or two earlier, but I think it was worth it. The James Rote Professorship paying for supplies and access to all the imagery that I used. The Ethel Miles Ocean Trust Award, CSU Coast, and the ML Wave Awards. Through my GFP, I was paired with Dr. Steve Lonhart at the sanctuary, and he was just gave me an abundance of knowledge and support during this time. Tons of people at CSUMB, my DSOs, Nick, Jose, Andrew, Sloan, all the people that helped me collect this data. Denise was with me for the first year or two, and if there was a photo of a diver and that diver wasn't me, it was Denise. We did 90-minute dives regularly. I was in my dry suit, and she wasn't, and she never complained. The folks at NOAA that helped me with this, they, like I said, lots of funding and a lot of resources and training, and it really, genuinely upsets me that these programs might not exist ever again. And then, of course, my committee members. James, just been wonderful throughout the years. I took scientific diving in 2016, and I knew that it was going to be important, but I didn't know that you were going to be important. Damn it. Amanda, such a warm, wonderful, insightful, thoughtful individual, and COVID sucked so bad, and I felt very detached from Moss Landing, but you never made me feel detached, and I genuinely appreciate that, and I literally think of you every time I see a sponge. Dr. Scott Hamilton, just so much knowledge, especially when it comes to statistics and the imagery, and I genuinely value all of the feedback you gave me. You held me to a high standard that has made me better, but I cannot forgive you for whatever accelerated hair loss I have experienced. Yep. Okay. The other people who, I don't have time to name, but all of my friends and Lyra. That was taken at Jedi night the other day at the soccer game. And my mom, who's just been so supportive my whole life. I couldn't have asked for a better mom. So thank you. I hope she's watching right now. Tambrina and our best friend, Lehua. You guys just every day coming home to you guys, or more likely being on my computer for the 8th hour straight and you guys come home to me, just made getting through this process so much better. So I can't wait to experience life with you guys on the other side of this degree. So thank you. And I'll take your questions now. Oh.
All right, excellent work.
Yeah, thanks.
All right, for the benefits of people online, any questions that we're going to field, we need to use the microphone. So just raise your hand and I will run the microphone out to you. So who do we have? Any questions?
I just answered them all.
Okay, hang on. Go.
Phenomenal talk. You definitely almost got me there at the end. My eyes are watering. I'm fighting it back, but that was really great. I just had a question about if you had any visibility issues. I'm not sure how close the camera is to the substrate, but did you scrap any data because you were just the software wasn't registering points, and how was that common, or was it only a few?
Yeah, so that is a good question. So I did not mention how far off the substrate I was. It was different for the two cameras. It was anywhere between 1 meter to 2 meters. The older camera system had a little bit tighter of a field of view. So if I go back, I used a narrower field of view for the, I might not find it, for the stereo video, for the habitat transition study, because we did have obstruction issues, whether it was usually low light or just a bunch of stuff in the water column. I'll look for it. But yes, that was an issue here. So this was only 1 meter to either side, or sorry, half a meter to either side, largely because of that. So yeah, it was an issue. The GoPro rig improved upon that. Really good low light performance. And I, most of the time, was not using lights. They were more trouble than they were worth. But those GoPros are really something else. That was a huge upgrade, and they're just getting better. So.
Don't hate me. I have one more because you swiped through and it reminded me. Would you be able to pull up your NDMS plot? I'm so sorry.
Yeah.
I know you're really proud of it because you probably put in a hell of work. So
Yeah. Well, so now you're gonna be mean about it, or okay?
No. So, no. I'm curious because you showed a lot of images of like the species in there. Like you saw purple urchins and you didn't see red urchins, but like, do you think that was being driven by like the number of them? Like your camera was just underestimating them? Like what?
For the
kelp forest community.
For these missing species?
No.
Or
just like some species that are in your green circle.
Yes.
Saw in your like.
Totally.
Were they just being like they were there, but like maybe just detected and all?
Yeah. So 3 of the largest contributors, Purpuratus franciscanus and crispatus. So we got a purple urchin, red urchin. So I mean, you saw those numbers. It was like 1,700 times more purple urchins found using the diver than the imagery. So definitely. And that's probably a little bit of the fact that there were fewer of them in the kelp forest. They're also hiding, and the algae, right? So kind of a triple threat there. The red urchins, similar story. And then, yeah, cryptic species with the masking crab. And then the other ones that are in there, abalone, there's just, no way for us to resolve them unless they're out and about in the kelp forest.
No, these are just so impressive. I had to ask a question.
Yeah. Yeah, thanks. Yeah, there's even one I didn't put up there.
This is the perfect slide for my question.
Okay.
Which is actually 2 questions. One is, I'm going to start with, they use a lot of these video transects in the deep sea.
Yeah.
And so you're showing that even using 2 cameras, you're missing certain things.
Yeah.
And so what from your study can we learn and actually change maybe our approach to sampling the deep sea? So that's for one question. But the second one was about the order of your 3 methods. And did you vary it, and what order did you do it in? Because on the urchin barren ones, where some of these algal canopy issues with visualizing the species, shouldn't be a factor, you're still seeing things like rock scallops and then these mobile species that might be, I don't know, maybe as we do multiple transects, usually rock scallops close up.
Sure.
And mobile species might hunker down.
Yeah.
So I'm wondering what was the order, and did you switch it up?
Yeah, so to your first question, I think using multiple different angles is, if I were to plan a project, exploring using multiple different cameras, whether or not they're going to be stereo capable, but approaching it from multiple different angles, because as a diver, if I'm flying over this podium here, I can look on the backside of it. One of the issues with the video approach is even the downward-facing perspective, I'm missing the backside of a lot of different things. And so if I were throwing cameras onto a ROV, it would make sense to me to have cameras in different parts on that vehicle. So if they were stereo capable, then that would be awesome. But I think that's a huge issue, is there's no flexibility on imagery approaches. A couple years ago, Cameron talked about 360 video. And this gets at that a little bit, but there's still a form of rigidity there that I think imagery will probably always struggle with. I've seen some really cool, there's some imagery inside coral reefs where they put it on a long snake and they snake the reef inside the coral. And I think that's a pretty cool application of it, but I think it's something that we're going to struggle with for a long time. As far as the second question goes with the order did not change. I'm having to go back now 4 years ago. The order was consistent. I did not intentionally randomize the order. So what I did intentionally do was the swath diver never went first. because of issues with stirring up the visibility or disrupting any of the creatures. So the transect was laid, and the first DOV pass was made. So certainly things like the rock scallops closing, those would absolutely be harder to resolve during a second pass. Even for the diver, for the swath diver, that would be harder, yeah. Something to consider for sure. I think Amanda may have mentioned something about that a few years ago when we were planning. Maybe I should have listened.
Hi, Travis. That was a great job. You clearly put a lot of work into this. I was curious if you'll entertain me if you would go to your flow chart, which was really awesome. By the
All
way.
right. You like that?
Yeah.
Cool. All right. That almost didn't make the cut because...
I'm glad you kept it.
Yeah, all right, sweet.
So I'm curious, if you had a situation with like Pisco or Reef Check, or a situation with like a team wanting to restore a kelp forest, how would you navigate this flow chart in those two different like scenarios?
Yeah, so I have thought about this a little bit because and I kind of mentioned it, I think a really significant use case for imagery in general, now that we're in this Urchin Baron, meta right now, is to go out and scan these with imagery, since nothing is in the way. Just go crazy, throw people out there, comb them back and forth. And then when we start these intervention efforts, then we have a really solid baseline. We'll still be missing some species, and I think that's where the issue then comes in, because once the kelp forest starts coming back, we're going to struggle to monitor it with these imagery approaches. So we're going to have to throw divers in there. And then there's the argument of, okay, well, what would the divers have always been finding that the imagery isn't? And then I think it depends on, the next question is, how important are those species? Like, do I really care that masking crabs are coming back or not? Or do I just really care that the urchins are no longer out and actively grazing? Is that the signal that I'm looking for to understand that the kelp forest is being rehabilitated? So I would immediately start with massive wide-scale imagery of these urchin barrens, and then probably weave in divers once the kelp starts coming back. I believe Reef Check they're taking cameras, like they're doing video transects on a lot of their dives now, and I think that's going to be really important when we are looking back at how effective, or just the general changes that we're seeing, the ebb and flow of these kelp forest cycles. So.
Thanks for entertaining me. That was
Yeah,
great.
absolutely.
Okay, any other questions for Travis? Nope.
Could you talk about the process of translating the video footage recorded by the survey divers into quantitative data? Who was doing that? What did that look like? And because I remember one of the limitations and benefits of the swath, the diver swaths, was that that specific diver had to have a lot of intimate knowledge of the study system.
Yeah.
So what was that translation process like for the video footage?
Yeah. Well, it was me. All of it was me. So I already knew the system. And I would spend enough time. I rarely went and jumped on the stereo video analysis. So I typically forget what it looked like down there, because that was something that I had to be considerate too, was I remembered what was down there, and I don't want to cheat the analysis. But for other applications of this, right now we're teaching in our fish ecology class, we have students identifying species that they never even heard of before in locations that they have never been to. But as with any imagery surveying technique, they can have a notebook or a guidebook right next to them. They can call up an expert to come, confirm their observations. So while I didn't necessarily struggle with that, this is a system I've been working in for a very long time. It is easily it's easy to remedy in application with people who are unfamiliar. It's one of the, I think, most compelling aspects of this approach, is that I can take any undergrad with any amount of experience, and I can sit them down in front of a computer, and I can stand over their shoulder and tell them that's what that is and that's what that is. Yeah. Did that answer your question? I hope?
Yes, and I guess
Okay.
a part two is when you would be, so 30-meter transect, and when you're after the fact, weeks later, processing this footage with your supercomputer mind, how long would it actually take you to finish coding the data for that 30-meter transect?
Oh, yeah. Well, that was actually a good slide for it. Let me go back. It would take me 181 minutes. It could take a long time, and that, one of my regrets was not doing a better job keeping track of this part of it, because it was a considerable amount of effort when I was counting every single urchin just along a 10-meter transect. And I acknowledge that I did not subsample. That's what people would do underwater because we cannot count 5,000 urchins on an aluminum 80, at 50 feet. That's not feasible. And so, there were similar issues that contributed to long times. But up to 3 hours for a 30-minute transect, that's a lot of time. But it's much cheaper time than spending it underwater.
Thank you.
Yeah.
All right, anybody else? All right, so I think the committee is going to take Travis away for a few minutes into the other room to do some stuff. So please come over and take advantage of the snacks. And let's give Travis one more round of applause.
The Zoom people don't need to hear whatever's about to happen.